Hi, everybody. Thank you again for taking the time to be here today. ECI Therapeutics has developed a proprietary platform that allows us to extract and purify and pathogen inactivate, meaning sterilize, allogeneic growth factors from pooled plasma from quite literally hundreds of individuals. We're able to start with platelet-rich plasma and extract from it the drug substance that we commonly know as safe and efficacious from autologous serum eye drops. However, we're able to do it in a way that's at scale, that is, makes the product accessible in ways that it is currently not today. So we're able to make it at scale, we're able to make it pathogen safe, and we're able to do it in a very consistent fashion by pooling from quite literally hundreds of donors. When we compare our product to what the treatments that are on the market today look like, we're really excited, and we're also really excited about the ability to help patients that are underserved by current treatments for neurotrophic keratitis. The initial indication that we're going after for neurotrophic keratitis is one in which there's 150 to 180,000 patients in the US alone, and there's about $90,000 to $100,000 of care tied up in each of those eyes. We're really excited to be able to implement a new technology, a new strategy for being able to treat those patients, and we believe that the revenue projections associated with that are going to be on the same ramp scale, if not faster, than was seen with Dompé's product. When we compare our product to autologous serum eye drops in vitro and in vivo models, we really see some exciting things happening. What you're looking at on the slide right now, on the left is our product, and on the right, serum eye drops, encouraging the cell proliferation and migration of corneal epithelial cells over a period of 48 hours. Over such a time frame, you can see with our product, we have almost 100% resolution of corneal ulcers or pseudocorneal ulcers. And we believe it's because we're able to concentrate and make very consistent the high levels of both neurotrophic and epithelial trophic factors necessary to drive the wound healing. While they're the same factors that are in the autologous products, we're able to concentrate them while not bringing up the levels of VEGF. We have a way to sort of keep that level lower in our process. Our product, compared to other treatments, is very stable. We have weeks' worth of room temperature stability data showing that the product can be shipped to a patient, and they can unfreeze it at their home and then keep it at room temperature for the entire course of treatment. We've done quite extensive animal modeling in disease-specific models. So we developed a model in combination or in conjunction with McMaster University that very much faithfully recapitulates the neurotrophic keratitis disease state. And we do this by both inducing a lesion on the center of the cornea and using a trephine blade to create a circumferential incision around the cornea such that the nerves are completely de-innervated. We then apply our product, control product, a recombinant nerve growth factor, and we look at how products behave. In all cases, and across the three endpoints that you see on the screen, corneal thickness, Oxford score, as well as percent lesion resolution, we're seeing that our product is outperforming both the control as well as neurotrophic keratitis' currently approved treatment, recombinant nerve growth factor. So this is really exciting for us. Percent lesion healing, which we think is the most relevant of the endpoints at the end of the 14-day course of therapy, we see approximately two-thirds of the animals treated, or two-thirds of the eyes treated with our product have complete resolution of corneal ulcers. For both the other arms, there was no more than 20% seen. And this is just sort of a after, comparing the control as well as our product. And you can see that with our product, there's really almost no staining, and there's no longer any incision that can be seen, but it can absolutely still be seen in the control arm. When you zoom in on that further, we see that we're actually achieving functional rescue from the neurotrophic disease state, and we can see actual reinnervation of the cornea, and you can see the cells of the nerves actually reconnecting over time in a way that we're really excited about, and we saw this much more in our product than any of the other arms. We are going to be applying for a IND towards the end of this year. We expect a Q1 launch of a combined phase 2/3 adaptive study design, approximately 60 patients with the opportunity to add more per sort of standard adaptive design features. And we believe that will be pivotal. Our lead investor for our upcoming 25 million Series A is the American Red Cross. They've signed on to be our lead investor. We have a signed term sheet with them, as well as a co-investor from the ophthalmology space that is going to co-lead. We're certainly still interested in having other ophthalmology participants, but we're really excited, and we think that we're going to close in the next 60 days or so. I'd love to hear from all of you or any of you that you have any thoughts or interest in either participating in the round or just general advice.